This is the Kratom Science Journal Club with Dr. Jonathan Cachet, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. It's accelerated solvent extracts of kratom leaves, evaluation of its cytotoxicity and antinociceptive activity. And this is a study by a team at University of Science in Malaysia. And this is basically about a new extraction method that they're trying on kratom. And this is probably, for labs and stuff. But since they said it's more cost-effective and less time-consuming, it might carry over into the commercial world.
Right.
Of making extracts. And there's just a whole an issue about extracts. There's full spectrum extracts, which are all the alkaloids in the kratom leaf that are extracted down. That's one type. And then there are extracts that are just mitragynine or just mitragynine and 7-hydroxy. There's a lot of stuff going on. I hear a lot of stuff about extracts in the kratom world, about how some companies are just extracting 7-hydroxy and then putting that extract in the kratom leaf to make it stronger. There's been some rumors of kratom vendors that are outsourcing their extraction methods to China, and they're coming back with traces of chloroform and methanol. Chloroform and methanol are two common extraction methods, and I think in everything, not just kratom. I'm just kind of learning about this. But what they're doing here is this study highlights, this is a quote, this study highlights the utilization of the green accelerated solvent extraction technique to produce a better non-toxic and antinociceptive active botanical extract of kratom. So it's kind of like they are using solvents. In this study, they use methanol, ethanol, ethyl acetate, and they just use a water for one, but they're finding that using this method, it's not resulting in any kind of toxicity being left over.
Well, and so the accelerated solvent extraction technique, ASE, has been around in the botanical space for a long time. And then the green at the front of that just differentiates essentially that we're not using chloroform or some other sort of environmentally toxic or contaminated chemical, which is presumably, the original. These techniques are used in almost all of botanical extraction and have been around as long as sort of botanical medicines have been around. Okay. But the use of like water, ethanol, and the ethyl acetate, those are at least compounds that are less toxic or potentially environmentally pollutants.
Yeah, and it says ASE and speciosa extract had a dry yield and showed a constant mitragynine content when extracted with organic solvents of different polarities. It only requires a shorter extraction time, 5 minutes, and a reduced amount of solvents, less than 100 milliliters. So that's why I'm thinking, I'm wondering why they haven't. I mean, it looks like this is the first time they tried this method on kratom, as far as they've said for the first time the ASC technique was employed to extract the kratom leaves. So I guess that's why it's not being used commercially.
Yeah, well, I think that they mentioned, I think maybe it's in the discussion or something, but they're essentially saying extracts used for preclinical purposes. And so the ASE, the accelerated solvent extractor equipment in labs, like I'm looking at one from Thermo Fisher right now, they say that they can accommodate samples of 1 to 100 grams and allows unattended extraction of up to 24 samples and uses less solvent. So your it's the scale is we want to just get some amount of extract to then be used like they did in the animal models or in the laboratory. And I'm sure that these accelerated solvent extractor equipment, is between 5 and 10 grand, if not more than that, way more than that. And so I think that they're primarily focused, like I mentioned, on the preclinical stuff. If you were wanting to do this in a commercial application, you would have to get a machine that is like, 100 grams is the max on this benchtop one from Thermo Fisher. You would want one that does like 100 kilos in a commercial setting. And not to say that they don't exist, but I think my point here is just that they intended it for preclinical models, and the lab equipment that you have to do for this particular ASE protocol is pretty expensive. I would say out of the reach of backyard chemistry or hobbyist chemists.
Yeah, and speaking of, I sent you a link to this guy that does, I think it's called Poor Man's Chemist or something.
Mm-hmm.
And he did an extraction in his backyard, which I would not try at all. But it was pretty interesting. But I think he did use chloroform. So what... Do you know a lot about chloroform? All I know about chloroform is if you're in a movie and you want to knock somebody out, you put a little bit of that on a handkerchief and run up behind them. But yeah. Apparently, I mean, is it dangerous to use in extractions for like consumable products?
Yeah, it's not good. I mean, it's not a very complicated molecule, but it's like similar to chlorine in some ways. And so in all chloroform packages, there's like hazardous labels, biohazard. I think that, if it doesn't just start messing with your like fundamental biology and causing problems, if it's the chlorine in there, then you're going to disinfect like your entire stomach and all the bacteria in there. And that also wouldn't be very good. I'm pretty sure, and I'm just scanning real quickly, some sites here, but I'm pretty sure that if you have too much chloroform, you can end up going blind.
Oh wow!
And that might be methanol. I might be getting it confused with methanol, but it's certainly, I think, was something that was used back in the day because it would get the people sort of knocked out. So, like in an anesthetic sense. But I think the margin of safety is much lower, and when they got better, better chemicals to be able to do that anesthesiology, they went ahead and did it.
Yeah, and like I said, I mean, this is just hearsay, but I've heard it from a few people that are paying attention to it, that there's certain companies that are supposed to be GMP. They're like larger vendors that are like outsourcing their extracts to China, and then they come back with toxic amounts of chloroform on them.
Chloroform as an incapacitating agent has become widely recognized, bordering on cliche.
Yeah, really.
Popularity in criminal fiction, and they're saying it's nearly impossible to incapacitate someone on chloroform with the whole rag over the mouth manner, because you have to inhale it for about 5 minutes in order to render a person unconscious. And I'm just reading Wikipedia here, but that's interesting.
Yeah, and there's a study in 2016 that University of Florida did, Dr. Sharma and McCurdy, that found that a lot of Kratom products, I don't even know if they're necessarily extracts. It might have been extracts at that point, but they were finding that there was an excess amount of 7-hydroxymetragine that occurs in the leaves, and that there was like a chloroform use to extract. I guess they would make an extract and then re-add it to the leaves.
It can be trace amounts left over from these type of extraction processes, and even in trace levels, it's not something that you want to be consuming. I mean, it's kind of like when we talk about heavy metals in the extracts, but the toxic solvents like the chloroform or the methanol can cause serious health problems. So it's definitely a problem to have those organic solvents inside of any sort of extract moving forward.
Also in this study, they were talking about other than alkaloids, phytochemical groups like flavonoids, triterpenoids, saponins, and glycosides. But I even looked up the definition of a lot of these, like flavonoids are in almost all fruits and vegetables, and they're responsible for the colors, and they're antioxidants. Saponin, they're referred selectively as triterpene glycosides, and they're bitter-tasting, usually toxic plant-derived organic chemicals that have a foamy quality when agitated in water. Glycoside is a compound found in simple sugar. With the extracts, they kind of measured levels of all that too.
I think in general, there's the alkaloids, and then most everything you just mentioned are like a type of terpenoid. Like if I'm remembering this correctly, a flavonoid is a type of terpenoid, but I thought it was more, color, yes, antioxidant activity, yes, but it also was like they named them flavonoids because it gave the sort of tasting versus a terpene that would be smelling. Flavonoids would be tasting. But yeah, so we're talking about, basically terpene-like compounds, and as well as simple sugars.
So would like an aqueous extract be kratom tea? Would that be because you're using water to extract stuff? I'm sure that's not exactly what they did here, what I just did with my tea this morning, but
an aqueous solution or an aqueous extract is essentially some sort of, it's when the solvent is water, and it has, so generally you can have like an aqueous layer and a lipid layer or an oil layer, and so stuff that's dissolvable in water would go to the aqueous layer versus the hydrophobic compounds, which would go into the oil-like layer.
And they also did methanol, ethanol, ethyl acetate.
Something at the beginning that I thought was interesting, I don't know if you've come across this before, but they refer to kratom as Korth, Korth. Have you heard that before?
Yeah, that refers to the guy who discovered it, Korthals. I think that's the full name, Mitragenous speciosa. Yeah. Gotcha. So Korthals was the name of the guy who actually, the first white guy who discovered it, who was a scientist, basically. I think he was Dutch or.
First white guy that wrote about it. It's so true. It's too true.
So so he slapped his name on it. He could have called it Mitragenous speciosa. Thai or something like that, or the country where it was from, or the name of the guy who showed him what it was.
right, yeah, exactly.
The guy that Dr. Korthals. So it says the toxic organic solvents, and they mean methanol and chloroform, are labor intense, have longer extraction times and low extraction yields. These problems can be overcome by using modern advanced green extraction techniques. We kind of talked about that.
Well, I think the labor-intensive part, I think what makes ASE like a core element of the ASE is that it's putting the the leaf matter or whatever you're extracting from under pressure. And so it's forcing the solvent into whatever you're extracting from. And so I would guess that, if you use the chloroform or the methyl the methyl solvents, if you put it under pressure, they would still be able to achieve a rapid extraction procedure. So it's not necessarily that, and there may be some other physical chemistry dangers of putting chloroform into a pressurized controlled vessel, but I think it's dangerous to handle, it's dangerous to store. It does take longer if you were just to let the plant material seep in it and stir it every few hours. But I think even more than that, the potential residue or trace leftovers that are in the extracted solution are the biggest problem with those solvents.
Yeah, it's almost like it should be a law that they only extract with, especially on a commercial level where you're trying to put out product. You're not in a lab trying to get the best extraction you can get. You're trying to sell stuff and do it as quickly as possible for as low a cost as possible. It probably might be good to not allow them to use methanol or chloroform.
It's kind of like cannabis extracts. I mean, right? You got to use either CO2, propane, butane, or a no solvents, a non-solvent like a pressure and temperature with rosin. I think that it may be the case that there are some plants or some alkaloids that just are not really able to be extracted using sort of like, ethanol or water. And I'm surprised with how I've seen kratom act in water, that water actually worked. I think it has to do with the pressure in there, and like forcing the solvent into the pores of the sample that is going to be extracted.
And that's the technique. I was just looking at what they're actually doing that's different, using elevated pressure to increase the contact area with analyte by forcing the solvent into the pores of the sample matrix. Analyte means the substance or the thing that they're analyzing, right? That's pretty much
that you're collecting. So the matrix is like everything that the target analytes are in. The analyte is what you're trying to recover. So in cannabis, like the sample matrix would be the plant and the flower material and all the stuff that goes into that, simple sugars, and the analyte would be THC. So in our case, it's metragenine or 7-hydroxymetragenine.
And it says the green solvents, water, ethanol, ethyl acetate, are more preferred by industries due to their low toxicity, safeness, and environmentally friendly properties. By that, they just mean like methanol's a fossil fuel. So is that what they meant by?
And you don't necessarily have to, like, so the safetiness is like how safe your workers are in just handling and using it. But then the environmental friendly has a lot to do with proper disposal procedures. And so with methanol, you have to put it in a special container, give it to a specialized trash, pickup company, or deactivate it in some way. Whereas you don't necessarily have to go through all those extra steps with water, ethanol, or ethyl acetate.
ASC, MEO, that's methanol, and ethanol kratom leaf extracts showed higher IC50 values against the kidney cell line and the liver cell lines. However, the mitragynine standard had lower IC50. So what do they mean by IC50?
Yeah, so they did the extraction and they measured it in multiple ways. So like different times, different extractions. Then they moved on to the cytotoxicity. So are these extracts showing any toxicity towards, and they use these liver cells as a moralized line of liver cells. And pull up exactly. What were the acronyms for those two?
HEK 290-293, that was the kidney, and HELA Chang liver cell lines.
Gotcha. So IC50 generally stands for inhibitory constant, but it's sort of like LD50, lethal dose 50.
Yeah.
And so it's basically the dose in which 50% of the cells that you're looking at are either inhibited or stop functioning or are destroyed. So it's generally, they go with the 50% range because they want to set up like a dose threshold that says this is where like it starts causing at least bad effects in 50% of whatever it's being exposed to. You definitely want to stay below that.
And it says, thus, all of the ASC kratom leaf extracts were found to be non-toxic, which IC50 is greater than 500 microgram per milliliter towards the liver cell lines.
And so they use that doxorubicin, which is known to be toxic to these liver cells and kidney cells. And so you can see that it was only like an 80 micromolar concentration of this chemical that ended up killing the kidney cells and the liver cells, whereas the extracts were all well above the 500 micrograms per milliliter concentrations, except for the metragenine, which seemed a little bit lower. But it seems to suggest, in terms of its toxicity, kidney cells and liver cells, that these extracts are even safer than pure metragenine itself.
Even the methyl one, but they didn't use chloroform in this one. Then they measured the extracts on these cell lines, and then they actually did them in Swiss albino mice using hot plate and tail flick tests, which we talked about these tests in other episodes. They had a similar onset time of antinociceptive action with morphine.
The similar onset time is essentially, so they administered these extracts And then for control, they had a vehicle, which is no active compounds, just sort of the saline solution that they're carrying, the extracts. And then they also used morphine. And so they dosed them. And then 30 minutes after the dosing, they started performing the tests. And in these tests, the longer latency until you like flick your tail or you lift up your paw or lick your paw. So however long, the longer you wait to do any of those things in response to the stimulus or the heat, presumably the longer or the better acting opiates is at killing your pain. So longer latency, more pain killing activity. And so, yeah, you can see that basically 30 minutes after, the extracts increased the latency in a way similar to morphine. And then after 30 minutes, morphine had the highest latency, and it continued that way over the course of 60 minutes, 90 minutes, and 120 minutes. Whereas, real close to that was the ethanol extract and the acetyl extract in terms of the profile and increasing the latency for those responses.
Okay, yeah, and I'm looking at table 6 now. It's like the compounds, and they measure all the compounds in the extracts, and it seems like as far as that goes, they're all pretty much the same. Like for example, my tragenine, it's 7.44 in the aqueous, 7.34 in the methanol, 7.34 in the ethanol. 7.49 and the ethyl acetate. Do you see anything there that's interesting or remarkable? It seems like every compound they have is pretty constant throughout, except there seems to be less of stuff showing up in the aqueous, like corinoxine. There's none of that showing up, and some of the other alkaloids and compounds.
So they tested a bunch of different variables in the extraction, and then they did the cytotoxicity by exposing to kidney and liver cells. Then they did the behavioral test with the tail flick and the hot plate. And then they also included this analytical chemistry section at the bottom to just perform a full characterization of the compounds that were in each of these extracts. And you're right, and I didn't really notice that at first, but regardless of the extract method, so this isn't the amount of metragenine found in there. They're giving the retention time in minutes. So it means that 7 minutes and 44 seconds in, the metragenine came out. And that would make sense because it's the same chemical compound. Okay. That's
I learned something there. Yeah. I'm constantly like I knew anything before. But another thing that I thought was interesting is they talk about ultrasonic assisted extraction, and that YouTube video, that guy had an ultrasound thing. Do you know anything about that? How does that work? It's just pretty interesting to me.
Yeah. I mean, so essentially it's like vibrating water at really high frequencies. So it's at 20 kilohertz to 2000 kilohertz. It's used in geology. It's also used a lot, males and females may be familiar with this, but it's also used to clean jewelry. Yeah. I mean, it essentially is a way to, the frequency and the vibrations get into like nooks and crannies and can shake off contamination or just like dirt on something really well. And in this case, the idea would be instead of using pressure to force your solvent into the matrix that you're trying to extract from, you're essentially shaking it in there with the ultrasonic assisted extraction technique. We also use that in a cannabis lab just as a way to sanitize and clean glassware and glass equipment, especially when you're doing analytical chemistry and you're running methanol, you're running ethanol through your machines or extraction machines. So yeah, it's just it's definitely what that guy had in YouTube too. They're not expensive to buy the little ultrasonic cleaners. You might even be able to get them cheaper if you search for one for jewelry rather than in a like chemistry lab or wet lab. But anyway, I thought it was interesting that they essentially mentioned that because of the ultrasonic frequencies used, it could result in undesirable modification of the structure of the compounds. And it makes sense, but I guess I had never really heard that from a chemist saying like, this is a big problem because if we use ultrasonic, it changes the compounds completely and their activity, and therefore we can't use it. I've never really heard that complaint before.
Yeah. And I've definitely remembered now because my wife takes her engagement ring to get cleaned like that too. Like a jeweler will have them in the back, and that's pretty cool. Maybe I'll get one. But they talk about other methods, sock slit and maceration. It said would take a longer time to extract plant materials. Sock slit, I assume that's a guy's name, Soxhlet, involves high temperatures and extracting, which may cause degradation. That makes sense.
Well, the sock slit too, like, if you've ever seen in movies, like glass apparatus in labs, there's a rotovap, which is classic, where it's like a big ball spinning in a water bath, generally at a tilt, and then a coil of wires where like things are condensing and then dripping down into a collection tube. And in some ways, the sock slit is the same. So it essentially like heats whatever you have in the bottom bulb, and it starts evaporating. And as it goes up through the glass, it reaches an area where there's cold water flowing around the outside of the glass, like a cold water sock. And so then it condenses that evaporation down into a collection test tube. And it's particularly, this method is particularly like well known and well used for extracting lipids from plant materials. And it has application. I don't know if there's one industry on Earth that hasn't looked to using extracts from plants in some sort of industrial way. So it's certainly a historically significant and tried and true method to extract compounds from any matrix, but namely plants.
So it says it can be generalized that ASE extracts are non-cytotoxic towards the liver and kidney cell lines. The results suggest that the synergistic interactions between active phytochemicals and kratom leaf extracts could decrease the toxic effect of alkaloids. Furthermore, reported that a high content of phenolics in extracts could support and increase cell survival in the HEK293 cell line. That's the kidney cell line. So it could support an increase in cell survival in kidneys, which is that in a certain context or just like in general, like it could promote cell growth or survival in terms of like if you're taking these other alkaloids, then your kidneys will be protected.
They're saying that the synergistic actions between all of the active chemicals in kratom leaves decrease the toxic effects of the alkaloids. Reported that high content of phenols in extracts could support and increase cell survival. So the higher number of phenolics in the extracts makes it less toxic. And the phenolics are antioxidant, and oxidation and oxidative compounds would lead to cell death, as it would like rip apart the cell walls. And so if these phenolics have antioxidant properties, the more phenolics you include in a compound, the less toxic they should be. Where we were talking earlier, like when it was the extracts, it was greater than 500 micrograms per milliliter. So high concentrations led to no cell death. But if you just have the metragenine in there, the metragenine, then there was like an IC50 where you started noticing decreased function in the cells or death of the cells. And so I think that demonstrates that including these phenolic compounds or other plant alkaloids in the extract makes them safer than just using pure metragenine.
So we're kind of getting into the entourage effect once again, which I kind of made a note for some other another quote here is like taken together the agonist, antagonist, synergistic interactions among alkaloids, flavonols, and other active constituents acting on multiple targets could have collectively exerted the antinociceptive effects. So even, I mean, that's completely different. That's talking about the pain relief in mice, but so that's another. But yeah, this whole cell survivor thing kind of suggests that there's the entourage effect once again is better than just the extracted metragenine.
Yeah, and they didn't have just pure metragenine in the tail flick or the hot plate test. I scrolled up to see that because based on what they're suggesting, the extracts would be much more effective at increasing the latency than metragine by itself, is what they're suggesting. But it doesn't seem that they did that test, at least not that I could see in the main paper. Maybe it was in supplemental. I mean, they really checked off all the possible interactions one could have: antagonistic, agonistic, synergistic. But yeah, all of that is sort of represented when we say the entourage effect.
And on this anti-nociception part, it said the ethanol leaf extract provides a better nociceptive relief in mice compared to the ethyl acetate and methanol extracts, despite their similar mitragynine content. This suggests the involvement of other bioactive phytochemicals besides mitragynine in the antinociceptive activity. But that's only... So why do you think like the ethanol extract behave differently than the other ones?
If I had to guess, I would probably say that because ethanol is a much broader solvent, so like it probably pulled through more of those other phytochemicals like through into the extract, so it had like additional phenolics or additional flavonoids, whereas like with methanol or with the ethyl acetate, they're a little bit more selective. I mean, broadly speaking, these are all, very general solvents. You can get into solvents that like just pull out single, families or even just single compounds. But with how ethanol is used in industry, like in cannabis, for example, if you're growing hemp to make CBD, you're using ethanol to extract it because you basically just want to get down to the CBD or the other cannabinoids. And so broadly, you can just use ethanol to wash all of those compounds through and then evaporate the ethanol and purify it that way. Versus like CO2 or butane are much more selective compounds. When you're running it, you're getting like the terpenes and the cannabinoids on the first or second pull through the matrix. And so, yeah, my guess would be it's because ethanol is a broader solvent that's pulling through more of those compounds.
So I told people we were going to do a thing about extracts, and I asked on Twitter, this is just not like at 10:30, but Drew Turner commented, and I always look to him for wisdom on just kratom in general, and does anybody have any thoughts on extracts in general? And he said extracts hold the most promise if standardized, and they hold the most problems if not regulated well. They will become the biggest issue and problem if a ban ever does happen. Extracts are kind of where you might fall into an expensive habit. The most promise is you're going to get the desired effects out of something that's carefully extracted. But if a ban happens, that'll probably be the most extracts will probably be the most likely thing that might be adulterated with fentanyl, kind of like we hear about heroin being adulterated with fentanyl.
It's like essentially the analogy between smoking cannabis flower and doing dabs. And so it doesn't really matter when it comes to this banning stuff, as it doesn't really matter what the pharmacology of the situation is. Like if they perceive it as being stronger and you're taking it in a way that is like if you're getting on a torch and now you're consuming cannabis with the torch, like it doesn't matter if it's the same compound. People just look at it like, what are you doing? What is this hard drug you're doing? And so he's right in that the extracts hold the most promise in that, like, if you're actually using it therapeutically, you're going to want to not consume 10 grams of powder. You're going to want to consume like a smaller amount to get the same bang for your buck. And so in a therapeutic way, and people that are using it to slide off opiates, using those extracts, personally, I prefer the extracts over the plant material. But you're right. You can then, it's stronger, it's easier to take, it's more expensive. So you could head down a more troublesome path if you're abusing it in that way. And of course, the regulators they're just hearing the stories. They're not looking at the science. And if they, this person took, 6 to 10 extracts in one day or something like that, and they got really sick, and now their kid's sick, and the mom's asking why the kid's sick or the dad, and they're saying, oh, well, you took these, I found all these extracts in his room. You know, it's sort of the same risk as vape cartridges or cannabis oil. It doesn't necessarily make pharmaceutical sense, pharmacological sense to differentiate between the two, but socially there's a clear differentiation. And so I think that's the risk he's referring to. I think he's dead on it. And you're right too, in that there's much more risk of contamination through the manufacturing process of the extracts versus just like grinding and capsuling up the plant material. And I think society has a way of accepting natural medicine, but it has to remain as a plant. It can't go into a liquid or something stronger. It's no longer natural at that point, even if the chemistry is identical.
As it is now, you hardly know what kind of strength you're getting or which alkaloids are more abundant than the other. I mean, if you get a full spectrum extract, then that's good, but sometimes you might get one with like unnatural levels of 7-hydroxy, and Kratom has such a wide range of effects, and you might want one and not the other, and with a ban, you're never going to know whether there's contaminants like chloroform or methanol in it, or B, whether somebody wants to spike it with fentanyl to make their product stronger, or C, if you're going to get more 7-hydroxy or more speciophyllene or anything else. So I think, regulation and keeping it legal is the way to go, especially for Kratom, which is such like a... It's just such like a... Even the extracts are kind of dynamic and...
rather benign. Yeah. I mean, it's similar to caffeine. But you're right. Yeah, I mean, there's like unintentional contamination where like part of your extraction solvents weren't fully evaporated. Like you should have spent an extra hour in the evaporation phase, but you wanted to get it out the door, so you cut it short, and now there's trace amounts of the potentially dangerous solvents in there. But then there's also room for intentional contamination where they're trying to like either avoid Kratom altogether and just putting in, we had that one that was a variant of tramadol. Or, even more dangerous, something like fentanyl, where they're trying to cut costs, yet have an effective product that people come back for. And regulation is the best way to solve for both of those things, especially with like good manufacturing practices and regulator inspections on the factory production line. Deregulating or making anything illegal is going to lead to more of those problems, because now there's no one checking. And in fact, they're off the grid. They're underground now at this point. So we don't even know where they exist. And they're certainly getting away with whatever they want at that point. So I think the comment and response on Twitter was dead on. I saw that he was involved in the documentary too. So I thought it was cool that he responded to us and threw that out there. He's definitely right. But I think we all agree that regulation is a far better approach than criminalization.
Thank you, Dr. Jonathan Cachet at jcachet on social media. Thank you, Drew Turner, for the comment. Please leave comments of your own anywhere. Like and subscribe, comment, rate, review. It really helps us out. And thank you for your engagement, all those of you who leave regular comments. The Kratom Science Journal Club is produced by me, Brian Gallagher, for KratomScience.com. Take care.